Nancy Webb, and I'm chairing the Vascular Discovery 2018 Scientific Sessions, which is being held in San Francisco this year. And we just wrapped up the opening plenary session for the meeting, which had a focus on precision medicine. And I have with me Dr. Kiran Musunuru, who organized this session. He is the editor-in-chief of Circulation, Genomic and Precision Medicine. Really interesting plenary session, Kiran, and why don't you tell me a little bit about how it came together and what the intent of the session was?
Sure. So what we really wanted to do was highlight precision medicine. You know, there's a lot of buzz about precision medicine. You hear it all the time in the news. The U.S. government is funding a very large program to recruit 1 million or more individuals from the general population, just kicked off actually this last week, to gather lots of data, genetic data and other types of data, in the service of better understanding the scientific and the clinical underpinnings of disease in a very big way, a broad way that's representative of all the different types of people who are in this country. But precision medicine means a lot of different things to a lot of different people. So part of our objective was to really bring in some excellent speakers who are working on various facets of what you might consider precision medicine. So the talks we saw in the opening plenary session this morning, we had a talk about using in medical or healthcare systems, using patient data, genetic data paired with electronic medical records in order to really be able to leverage the health of tens of thousands, hundreds of thousands of people and be able to use their health status, their data, their diagnoses, all in a de-identified way, of course, for research purposes, and all with consent, of course, but to be able to leverage this large trove, if you will, of data in order to better understand the relationships between genes and diseases. Another talk we had this morning was on the use of induced pluripotent stem cells, so cells that you can make from individual people, taking their blood cells and then turning them into stem cells, and how they share the same genetic makeup as the people from whom they came, and so they can be used as little proxies or avatars. And so you can study them in the laboratory without ever having to touch the person again, and then be able to, for example, treat them with medications and see how those cells in a dish respond, and then be able to extrapolate that and make predictions as to how the person from whom those cells were made will respond. So a way of being able to do, you might say, clinical trials in a dish. Yet another talk was on what we call pharmacogenomics, using genetic information to predict people's responses to medications. For example, antiplatelet drugs that are used to prevent clotting after a person suffers a heart attack and has a stent placed in their coronary artery in order to keep that artery open. A very important thing to do is to keep it open and prevent closing off by a new fresh clot. And so given a very powerful blood thinner, but different people metabolize or process that blood thinner in different ways, and we now have a much better understanding of how these differences at the genetic level affect the response to the medication, the propensity to clot, whether they're on the medication and responding it too well, or if they don't have the optimal genetic variants, maybe don't respond to that medication as well. And then that actually translates into a difference in outcomes and whether they actually have another heart attack down the road or not. And now that we know this information, we can make better predictions as to which patients should receive the standard blood thinners versus those who probably shouldn't receive the standard blood thinner, because we can predict that they're not going to respond well to that and then prescribe alternative medications. And so you can see these are somewhat different areas, but they all impinge on precision medicine, on the way that genes and lifestyle and behaviors and environment and so forth interact, really to make each person unique. And you know, this opening plenary session was just the beginning. We actually have a whole full day of events planned. We have another session that's also going to feature speakers about who are going to be speaking about the different flavors of precision medicine, for example, using machine learning to better understand the various types of heart failure that patients can experience, better understanding how large collections of individuals in research studies, that number as high as millions of people, can better inform our understanding of disease and better inform the medications that they will best respond to, both in a positive way, by which I mean efficacy of treatment, and potentially in a negative way with respect to adverse effects. And then our sort of ulterior motive was to get these really great people here in one place at one time to put their minds together and have them serve as a writing group to put together a white paper to really outline the current status of precision medicine as applies to cardiovascular disease, but also to forecast where it's going to go over the next few years. And even more than that, I think set some priorities or research directions in which precision medicine to go to really help steer the field, if you will, in productive directions that will ultimately favorably impact patient care. And so we're having those activities, not just the talks that they're giving to inform the attendees at this conference about precision medicine, but also having things going the other way to help nucleate some discussions about precision medicine in future directions, and not just as a group of individuals, sitting in a room off by themselves and thinking about precision medicine, but actually getting opinions from people here at the conference. So at lunchtime, we have an open town hall where anyone who wishes to come can come and participate. The writing group members will serve as sort of a panel, and the topics will be discussed, but anyone who wants to weigh in, who wants to give an opinion or suggest new topics or new priorities, will be welcome to pitch in. And we're, in a sense, trying to crowdsource the generation of a white paper that will ultimately be published in my journal, Circulation, Genomic and Precision Medicine, and be something that everyone in the field can point to as, this is where things stand in 2018 with respect to precision medicine, and this is where we as a community would like to go forward.
I'm really interested in hearing what comes out of the town hall meetings, because one of the things that you haven't addressed is some aspects of it. For example, concerns about privacy issues, the cost of this new and emerging kind of medicine. And I'm wondering if your panel of experts will be dealing with any of these issues.
It's certainly something that always comes up when talking about big data and talking about taking patient records, right? I mean, it's their medical data, and you absolutely want to give it the most rigorous, appropriate protection so it's not used in any way that could be potentially detrimental. So privacy is a big concern. Consent is a big concern. These are always issues that arise in the discussion, and it's certainly something I would expect the writing group to tackle in a significant way in the generation of the white paper.
Thanks, Kiran, very much. I think this is a very exciting aspect of our meeting this year.
I just want to add one more thing, if I may. One very important motive behind our session this morning was to arrive at a definition, a shared definition of precision medicine. Again, recognizing that it means so many things to so many different people. It's sort of a fuzzy word, has the flavor of a buzzword that everyone kind of understands what it means, but no one can give a single perfect definition to. And so we made an attempt to come at that one consensus definition. So what we did is we had polled a large number of people and winnowed down a list of suggested definitions to 16 definitions, and then we had some fun with it. We had a March Madness style single elimination bracket where we put two definitions head to head in matchups, and then had several successive rounds, ultimately culminating in a final four, and then a final championship round, and then finally a single winner. And I'll read you the definition that was chosen by the audience at the opening plenary session here at the Vascular Discovery meeting: a clinical and scientific strategy to optimize patient care by individualizing diagnosis, treatment, and prognosis, facilitated by ongoing advances in molecular and clinical diagnostics. And I know that's a mouthful, but if you unwrap it and think about it carefully, I think it's actually right on, and I'm actually very pleased with the result of our little competition.
Great. We'll send a note to Webster's Dictionary. Thank you, Kiran.